Hello, I'm Tim Stout. This is a schematic diagram of a single experiment that kicked off an entirely new field of science called abiogenesis. Abiogenesis is the theoretical conversion of raw, non-biological chemicals into living cells through natural processes. This experiment is called the Miller-Urey experiment. In it, a spark supplies the energy to convert certain raw materials into over 100 different compounds. They're removed at the trap here at the bottom. It formed these in only a week. Some of the chemicals that were formed are called amino acids. These are used to make the proteins so important to our lives. However, most of the chemicals formed were not amino acids, but were aggressive contaminants. These contaminants would prevent the amino acids from ever stringing together to form proteins. This experiment actually demonstrates certain inherent behaviors common to natural processes in general. They effectively make a natural origin of life impossible. 1. A raw energy source acting on naturally appearing chemicals can turn them into many kinds of new chemicals. Some of these might even be incidentally useful towards life. However, most were aggressive contaminants. The contaminants prevent the amino acids from forming proteins. No proteins were formed in this experiment. 2. The experiment clearly shows that there is no means to restrict the broad range of new chemicals naturally produced to make only those that are needed with the purity required for success. 3. The laws of randomization and entropy suggest that this problem is inherent to all natural pre-life processes. This appears to be confirmed by observation. Unless and until a scientist can provide observable evidence that these assertions do not apply, abiogenesis is a closed field. It is impossible. This experiment shows that abiogenesis cannot get out of the starting blocks. It cannot even take the first step successfully in the race towards life. This experiment has had many variations. Different starting chemicals, different energy sources such as UV light or high temperatures, and different environmental conditions have all been tested. However, none of these experiments offered any mechanism to restrict the output to only what was needed. Predictably, they all failed for the same reasons that the Miller-Urey experiment failed. Incidentally, I discussed these steps in thorough detail in a highly technical free research article available online at this URL. The article surveys the entire field of abiogenesis and shows how the things we looked at apply to every tested step in abiogenesis. This initial step, the provision of amino acids, is the simplest step of abiogenesis. It fails miserably. If the processes available in a pre-life scenario cannot even do such a simple task like this successfully, it is ludicrous to expect them to do extremely complicated ones. Besides, what chemicals would the later ones have to work with? Almost every encyclopedia article and every introductory biology textbook section discussing the origin of life will discuss this experiment. Sadly, they pretend it illustrates how a natural origin of life could happen. In truth, this experiment gives strong basis to understand not only that it is impossible, but also illustrates principles showing why it is impossible. The late Carl Sagan used to have an extremely popular television program on PBS called Cosmos. He actually reperformed this experiment and showed the results on YouTube. In the next video, we will examine what he posted, but from the perspective presented here. I think you will find this discussion interesting, to say the least. Please click the subscribe button on your screen if you want to keep up to date on these videos as new ones are posted. Also, please tell your friends about this channel. Thank you.